I want to talk to you about some of the recent progress in our lab. Actually, try to understand the metabolic challenge in especially as a therapeutic targets. And um, I want to first thank our team. And as some of you know, I recently moved to Singapore from many years at UCLA, and I really appreciate during this time our team members actually really working very hard, allow me to actually share with you some of this progress. So we know that metabolic and heart failure are both very, very complex diseases, many factors actually involved. And they are also incredibly connected. If you see the heart failure, much of the risk factors actually are metabolic conditions as well. And we know heart is very important, also metabolically demanding organ. It's not surprising. If you look at even from all the Asian cultures, heart is really vital. And I actually did not appreciate until I became a little bit more kind of as a teacher, when I teach cardiac physiology, I actually learn from Professor Tan's textbook. Is really the heart is amazing consumer of energy, and each day a human heart we have to feed the heart with 6 kilogram of ATP. If you think about, that's really a huge amount of energy actually being consumed. And so the amazing thing again is that, as I said, heart disease development hand in hand with the progression of the metabolic disorder. As we see that in the world, cardiovascular disease actually represents 1 out of 4 deaths, but also globally, the obesity actually also increased. And these two things actually become, we call it the super tsunami. They essentially feed each other and start to really have a very significant unwanted impact. Particularly in the area of a heart failure with preserved ejection fractioning. As we indicated in this white paper from NIH organized by the National Institute of Health, the heart failure with preserved ejection fractioning is now a major public health, actually the greatest unmet needs in modern day cardiology. And so what is this? And turned out heart failure is with preserved or so-called HAPF is really complex diseases. It's not just one disease. And the most challenging find is that almost all the classic therapies actually failed in large cohort of standard therapy actually showed those particular patients actually are refractory to those standard therapy. And the only thing that worked right now is actually the clinical trial from SGLT2 inhibitor. As we know, originally it was a diabetes drug, right? So it turned out that it's the metabolic that actually underlying perhaps the disease organ pathogenesis. And again, there are many hypotheses being actually proposed over the years and talk about the complexity of the disease, more than just in the heart, but also vascular and the metabolic. But it's inevitably always involve a very strong metabolic component. And so as we see that in the preclinical models, many of those models actually will involve some aspects of metabolic disorder. And so we took this idea actually pioneered by Joe Hill and many other researchers to think that maybe there is a combination or a synergy between cardiovascular risk factor like a pressure overload or like hypertension and the metabolic disorder. So we designed this study really to understand what is the synergy, or what's the potential emerging new properties from a hypertension-induced heart failure versus a obesity, diabetes-induced cardiomyopathy. And so what we have done is that we designed this 4 cohort of studies in mice. One is the traditional pressure overload in the C57 black lean mice. Those are typical heart failure with reserve ejection fraction. And the other one is actually in the OBV mice, which is actually a typical diabetes, obesity mice. We apply the same stress. Because of the sensitivity of those mice, we actually apply slightly less pressure gradient. So using the 25 grit needle. And in those conditions, after 10 weeks, you can see that in the gray bars, most of the uh, uh, C57 black lean mice developed classic heart failure with reduced ejection fraction. Their ejection fraction is going down, and their many of the other parameters, including the hemodynamic parameters as well as the cardiac remodeling, actually will show the similar sort of impact. But if you look at in the OBOB mice, and turns out the same level of pressure overload at the same time, we never see any ejection fraction reduction. However, when we look at all those diastolic parameters, they indeed develop much worse in terms of diastolic function, indicating there's perhaps a more diastolic contribution to their heart failure. And indeed, they are absolutely heart failure. They actually cannot exercise. They bear all the symptoms of a classic heart failure. If you look at cardiac pathology, they also have fibrosis. They have inflammatory markers that are uniquely induced. So clearly, these are not necessarily better. They are actually as bad as the other heart. They just have a very different sort of features. So among these 4 cohorts, we decided to have a little bit better understanding. And since they are clearly in the same genetic background with a different treatment, we can actually hopefully using our molecular tools to study that. And one of the tools we apply is the single cell transcriptome studies, where we can actually allow us to study the different cell population contribution and changes. And it's a very extensive study. Today I have time to actually share with you what we have found from just the cardiomyocytes. And so you can see that if you take all the cardiomyocytes from the single cell studies data, and you'll see clearly that from the normal heart, they developed already a very different state under this diabetic obesity condition. And plus the pressure overload, you can see that they end up in a very different domain, right? And so this is the typical half ref, and here is actually the sort of a half path in between, quite more heterogeneous than the half ref. And if you look at the gene expression, differential gene expression, what really matters between those two groups, and what we found, it turned out to be insulin and the glucagon signaling. Very expected, but also somewhat unexpected. And if you look at those genes, you see that in the wild type animals, and the pressure overload induce one direction of changes. You can see the color change. And but if you look at in the OBOB mice, those changes, same molecules, exactly go to the opposite direction. So indicating that they must have another underlying mechanism to promote or those metabolic changes. They are all insulin resistance related, glucagon signaling related. And so we know how to make the insulin sensitivity improved, and that is actually what the SGLT2 is working. So we thought this could be a very interesting underlying mechanism for how the SGLT2 actually really works for this particular set of human heart failure. But also, we know the glucagon signaling, in theory, usually works in the liver. It's very little study actually done in the heart. But just a few years ago, we actually, it's by almost accident, we actually studied the glucagon receptor signaling in the cardiomyocytes. And turned out, when we utilize a glucagon receptor antagonist, they actually conferred very strong cardioprotective effects. And indeed, when we apply this glucagon receptor antagonist, we can essentially block the setup or starting or initiation of the diastolic dysfunction. But you may ask, what is the underlying mechanism? Because indeed, the glucagon receptor, as we expected, has very potent impact on global insulin resistance. And you can see here, the glucose tolerance test, the glucose clearance is much improved by the glucagon receptor antagonists. That's true in human. However, we use the glucagon receptor cardiac-specific knockout mice, and we actually see very much the same. They actually, this genetic knockout mice only happen in the cardiomyocytes and do not have a global impact on the insulin signaling. And yet, when we applied both the pressure overload and the high-fat diet, you can see the diastolic function is much improved. And so, on the other hand, beyond the glucagon signaling, we also found the other kind of a signature, particularly from the metabolomic studies, is the branch amino acid change. It's quite significant associated with the HAPF. And we know that branch amino acids, there are essential amino acids, and we don't synthesize them. They serve more than just the protein synthesis, but also very strong nutrient signaling. And over the years, we realized that the BCAA is actually tightly regulated in individual, in human, and as well as in animals. And we're lucky that early on, we discovered some of the key regulators, especially through the branching ketone acid dehydrogenase complex modulation, to maintain the homeostasis of this BCAA metabolism. And turned out, genetically, if we knock out the BCKDH activity in the heart, you can have a very immediate and very profound impact on the heart function. And if you knock out the kinase, which is actually a reverse or inducer of the metabolic pathway, only in the heart, you can actually protect the stressed mice from death. And so this can be applied to pharmacological intervention, because the BCKDK is a kinase where we actually lucky enough to get a small molecule to highly specifically inhibit it. And indeed, early on in 2016, we actually published this study on the BCAA targeted therapy for HFrEF. However, so this is just a follow-up study. And however, as I indicated, that based on the serum marker metabolomic studies we have done, we found that a major module that associated with all the adverse outcome linked with this people of HAPF turned out to be this yellow module. And when you look into what is the yellow module composition in the metabolites, they are very clearly enriched with branched amino acid. That's the valine, leucine, isoleucine. So those are, completely unbiased study, and yet still point to the BCAA impact in the HAPF. And so we applied the same approach using the BD2 or branch ketone acid inhibitor. And in a established model of a HAPF, this is actually the same as Joe Hill's Nature publication, where using both high-fat diet and LNM, you can produce a very specific, profound impact on the diastolic function without affecting the left ventricle ejection fractioning. And you can see that in this model, if we apply the BD2, you can actually significantly improve the diastolic dysfunction, as well as some of the marker genes, and which I don't have time to show. And so I'm very happy to see that today, this year, Pfizer actually took, based on our study, actually developed this HAPF new drug, and this target is exactly the one that actually we discovered BCKDK in our approach. And so today I share with you these two interesting metabolic pathways. One is the glucagon receptor mediated signaling. The other one is actually BCAA. We believe that they may represent additional, in addition to the glucagon, the SGLT2 inhibitor, maybe represents another complementary therapy for HAPF. This is actually quite relevant because the SGLT2, both SGLT2 actually SGLP inhibitor agonist, they all increase the glucagon receptor, glucagon secretion in the patients. So maybe the combination therapy will be actually more efficacious. And so with that, I want to thank our collaborators and the funders, and also since I'm at DukeNUS, I have to put on this hat that we're recruiting faculties. So thank you.